right? Glucose, this is our, remember, 6 carbons. Keep track of the carbons here so you know where, every time we lose a carbon, that means we have to produce a carbon dioxide as waste. Okay, because carbon dioxide is very low energy, that means that our cell has extracted as much energy as it could, and it's producing carbon dioxide. Okay, so glucose, we start with 6 carbons. Remember, this came from all the way back here. Okay, if you're in a plant, you're using the same glucose you made in photosynthesis. Okay, this is our process called glycolysis. So I'm going to write that, maybe in a little bit different color. Okay. Glycolysis.
Yeah.
Okay, we make pyruvic acid. 3 carbons. Let me just go ahead and circle these. Now, this process of going from glucose to pyruvic acid, it costs 2 ATP. So we have to actually pay 2 ATP, but we get 4 back. So I'm just going to write the net total that we get from glycolysis, which is 2 ATP. So we go from, so I'm just going to say 2 ADP plus phosphate to 2 ATP. So we've made 2 ATP. We've also released some high energy electrons in this process. We have to capture them. So we capture them in our electron carrier in cell respiration, which is NAD+, and that gets turned into NADH. So, ultimately, at the end of glycolysis, we have, two pyruvic acids, 3 carbons each. So we haven't lost any carbons yet, so we don't have any carbon dioxide emitting. We've got 2 ATPs, and we've got some NADH. That's glycolysis. Now, at this point, we have our 2 options, right? If we're in the presence of oxygen, we can then go through cellular respiration. However, if we don't have oxygen, we go through fermentation. So I'm going to do fermentation first, because it is the simpler of the 2. So at this point we have our dividing pathways, fermentation or cellular respiration. Now fermentation is this way. Okay, and there's 2 types of fermentation. There's 2 types of fermentation. We have alcoholic, and we also have lactic acid. Okay, this one releases a carbon dioxide. This one does not. Now there are a lot of steps involved in this that I'm not going to go into. Just need to know that there are 2 types, and this is the product of the 2. However, this gives us 0 ATP. We don't get any ATP here. But what's the point of doing it? It's to take NADH, turn it back into NAD+. The NAD+ can go back, so we can keep doing glycolysis. The whole point of this is so we can still scrape out this measly 2 ATP, right? So if we don't have oxygen, we can still at least make a little bit of ATP to accomplish something, at least in our cells. Right? If we didn't do fermentation, what would happen is we build up all this NADH, run out of NAD+, and we'd be kind of out of luck. We can't keep doing glycolysis unless we have NAD+ available. Okay, so those are our 2 sort of fermentation pathways. And then, so this is without, let me just keep, without oxygen. This is with oxygen. I don't know if I have a whole lot of room to fit this in, but I will try. Okay, we're going to go, so if we have oxygen, these pyruvic acids, they're going to go and enter the mitochondria. Okay, now remember, this is taking place in the mitochondria. So I'm going to draw the mitochondria here. What do we do? We got...
Mr. Jones?
Yeah.
Then where does fermentation happen?
Fermentation happens in the cytoplasm.
Okay.
Just where glycolysis happens.
Okay.
Yep. Okay, so
If we do fermentation just to get rid of NADH, what's the point of taking that?
What's up?
Okay, so if we use fermentation
Yes.
to get rid of NADH and turn it back into NAD+,
Yes.
what's the point of making it in the first place?
What's the point of making this in the first place? Because if we are going to do cellular respiration, we want to utilize, we can't, because electrons can't be free floating. If we generated a high energy electron, it can't just be in the middle of nowhere by itself. It has to bind to something. So that's what NAD plus is, it's an electron carrier. So we generate, in this reaction here, when we split glucose, we give off high energy electrons. They need to be captured by something. NAD plus is that something. So we have to regenerate it here if we want to keep doing this. We can't just let them be floating around because they're damaging if they try to bind to other things. Like an electron flying around randomly is dangerous because it's going to want to bind to something and it could disrupt whatever that something is. So we want to make sure that we capture it in something that we know isn't going to cause damage. Plus, it's got high energy and we can use that later in the electron transport chain.